My name is Dr. Jodie Rummer. I am an associate professor at James Cook University within the Australian Research Council Centre of Excellence for Coral Reef Studies. I'm also the chief investigator of the PHYSIOSHARK research program. I guess the overarching theme of the PHYSIOSHARK project is to understand how current and future stressors will affect newborn sharks or baby shark populations, and how that will play into the health and viability of adult shark populations. And this isn't really just targeted at Moorea. Moorea is an amazing field site, amazing place to work, but we're really using Moorea and French Polynesia as a starting point and as an example for what can be done and what can be understood on a global scale. Another great thing about working in French Polynesia is that it's a shark sanctuary. So we would think that this is kind of the best case scenario for sharks. They're overall protected from being targeted in the fishery. So they have one of those major stressors removed from their lifetime at least. But we know even the best protected marine parks, the best protected shark sanctuaries aren't immune from climate change. And so that's something that we really still need to understand.
First thing in the morning, if I have any sharks in the lab acclimating for any particular experiments, first thing I'll do is go check on them. If they've had a meal the night before, make sure everything's been eaten, or I'll clean out the tank. If not, for the animals that are acclimating under different conditions, make sure that everything's working properly, the conditions are stable, and I'll just generally check the water quality and make sure the tanks aren't too hot, that there's a sufficient amount of oxygen in the tanks as well, and that they're not too salty or too dilute. So in the afternoon, I typically prepare for any field work that's going on that night, or any laboratory experiments that I'll be conducting either that night or the next day. So our field work typically involves catching sharks, newborn sharks with gill nets. Meters from our car, we can start fishing and catch these animals. So we catch them with a gill net. It's about 1.5 meters tall and 50 meters wide. And so that net gets dragged out from shore to about 50 meters. Now by 50 meters offshore, the water in some sites is too deep for the little sharks to safely swim, because larger predators might be able to get to them. So the net is perpendicular from shore, and it creates a barrier where an animal cruising up or down the coastline might potentially swim into the net.
So in the field, we have a suite of measurements that we like to take. And we have not only the 4 years of data from when PHYSIOSHARK started, but we also have some historic data from other projects that have gone on here on the island. And what we like to understand is maybe some of these growth rates of these newborn shark pups in these areas, and if we are recapturing them. So we've got a tagging process that we will use as well, so we can measure the sharks, tag them, and if we get them back, we can know how much they've been growing over that period of time. We're also taking a small biopsy from the end of their fin that we save, and we can analyze for DNA. We can understand how related some of these shark pups are, and also which individuals are their parents that are out on the reef. So we can understand sort of a familial relationship of all of the sharks that are living around this island, and how that's connected to these different potential nursery areas around Moorea.
I'm running a series of experiments this field season that are largely laboratory based. So these laboratory experiments involve bringing sharks into the laboratory and then acclimating them at different temperatures that these animals currently experience, as well as a temperature that these animals are predicted to experience routinely at the end of the century under current climate change scenarios. So that first performance test is conducted at 2 weeks to look at the shark's aerobic scope. Again, at 4 weeks, once the sharks had a bit longer to acclimate, I'll measure aerobic scope again using the exact same performance test. And then there are 2 additional tests we'll look at as well. The first is a hypoxia tolerance test. Hypoxia simply means that the oxygen level in the water gets really low. So this test is a behavioral-based test to look at how low can the oxygen get before we see signs of distress, before a shark does not want to be in this water and has got to leave. So the second test is a thermal tolerance test. And this is trying to understand, well, what's the hottest temperature that these sharks can thrive in their environment? At what point does it become too hot?
So the overall goal of the PHYSIOSHARK research program is to understand how stressors that are human-induced, such as climate change, perhaps especially climate change, are affecting shark populations. And we're particularly focusing on shark populations in the tropics. We think that tropical shark and other fish populations may be most vulnerable to climate change because they are living in and maybe adapted to very narrow ranges of conditions, such as temperature. They don't get a lot of seasonal variation in what they're experiencing. And so increases in temperature that are coming with ocean warming, on top of that, could be quite detrimental to these types of populations. So that was really where the focus started, to understand how shark populations around the island of Moorea would accommodate some of these conditions that are happening up through the end of the century. If we can set an example, not only here in French Polynesia, but maybe on a global scale, of how great a shark sanctuary can be, but yet still try to understand how these additional stressors that are coming with climate change can affect these sharks, maybe we can do a bit of a better job of managing how much stress these populations will get as babies and be able to better protect them as adults.